या फिर इसको दोबारा कर दो, लाइव को ना इसको डिलीट करके एक और बार कर दो, दो बार restart कर दो। दोबारा से ठीक है. Welcome everyone to the revision series. So for today we are going to do genetics in one shot revision session. So let's start with genetics. Now the person who introduced genetics and is known as the father of genetics is Gregor Mendel. Mendel for his experiments chose pea plants, Pisum sativa, and he concentrated on 7 characters of the pea plant. He performed different hybridization experiments with respect to these 7 different characters. Mendel performed 2 types of horses, monohybrid cross and dihybrid cross. Let's first do monohybrid cross. The word mono means single. So this particular cross was done in respect to a single character or a single gene. Okay. So for monohybrid crosses, the parents that were selected were pure line, simply referring to the fact that they were homozygous. The parents selected were homozygous. Now, for example, let's say the single character gene selected was height. So there can be 2 traits for the character height. The plant can either be tall or it can be dwarf or simply short. Okay, so the parents selected were homozygous dominant, that is tall plants, as well as homozygous recessive, that is short plants. So when a short variety was crossed with a tall variety and both of them were homozygous, what Mendel observed is in the F1 we were getting heterozygotes. We were getting heterozygotes, that is one capital T and another small t. So these plants, they were showing one particular phenotype tall. Okay, so the short phenotype was not observable in the F1 progeny. All of the progeny were heterozygotes and all of them were tall. They showed only one phenotype. Further, when Mendel performed crosses with the F1, he inbred F1. As a result, the F2 progeny that we got we got 2 heterozygotes, one homozygous dominant and one homozygous recessive. So the ratio that we are getting, the genotypic ratio, is 1:2:1. The genotypic ratio of monohybrid cross is 1:2:1. Now what is the phenotypic ratio? The phenotypes observed were tall, tall and dwarf, right? So both the homozygous dominant as well as the heterozygote plant showed the same phenotype, that is tall, and we also received dwarf plants in our F2 generation. So the phenotypic ratio would be 3:1. 3:1. So these are classical Mendelian ratios that most of the traits do follow, but there are also genes or characters that do deviate from the given genotypic as well as phenotypic ratio. Now based on these monohybrid crosses, Mendel also gave certain rules and laws. Law of dominance and law of segregation. Law of dominance stated that in a heterozygous condition, the allele that expresses itself is dominant. while the other allele that is not able to express itself in heterozygous condition is the recessive one. Okay, so this was the law of dominance. One particular trait expressed itself while the other one was not seen in the F1 gene, that is in the heterozygote condition. Second is the law of segregation. The law of segregation states that during gamete formation, the alleles are going to segregate. So for example, we have a heterozygote individual. When this heterozygote individual is going to form gametes, both of the alleles, they are going to segregate in the process of gamete formation during meiosis. So this is what is meant by law of segregation. Okay. So Mendel said that genes regulated characters. One particular gene is responsible for one character, and these genes, they are discrete units. Well, he called them as factors. Later we call them as genes. Now genes or factors are discrete units and they do not blend. That is why we do not get an intermediate phenotype anywhere. The plants were either tall or short, and an intermediate phenotype was not observed. Now genes have alternate forms. These alternate forms of genes are known as alleles. Alleles. Now the dominant allele is shown with represented by a capital letter, whereas the small or recessive allele is represented by a small letter. Okay. So there are genes. Genes are responsible for a character. For example, if there is a height gene, it is responsible for the character height, right? And that character height can have two traits. Either the person can be tall or they can be short, depending on which the alleles are there. Alleles are responsible for one particular trait. For example, here the dominant allele is responsible for a particular trait, let's say tall, right? Whereas the recessive allele is responsible for a trait short. Okay. And when both the alleles that are present in a particular individual, they are different, that is heterozygous, and when they are same, that is homozygous. So this was the monohybrid cross. Let's quickly move on to the dihybrid cross now. Now in monohybrid cross, we considered one gene and two alleles. In the dihybrid cross, we are going to consider two genes. So in the dihybrid cross, Mendel concentrated on two genes, that is seed shape and seed color. Okay. He selected these two genes, and again the parents that he selected were pure line, simply referring to the fact that they were again homozygous. The parents he selected were homozygous for either traits. So he selected a homozygous dominant parent, and another parent was homozygous recessive, and he crossed them. You can simplify dihybrid crosses by viewing it as a monohybrid cross, right? So simply view it as a monohybrid cross and try to solve this. So if we cross a dominant homozygous with a recessive homozygous, we are going to get heterozygotes, right? Similarly, for the other pair also, we can solve this. So simply in the F1, we are going to get heterozygotes, right? We are going to get heterozygotes. Now, this particular parent was round and yellow, whereas this one was wrinkled and green. And the F1 resembled the dominant parent, showing only the dominant alleles, right? This is also round and yellow. Now in the F2 progeny, we observe another phenotypic ratio. What is this phenotypic ratio that we observe? We are getting a ratio of 9:3:3:1. This is the phenotypic ratio. In the F2 progeny, we get a phenotypic ratio of 9:3:3:1. Right? So these ratios, they are very important, and all the genes that follow Mendelian inheritance, they show these particular ratios, as well as they obey the particular rules and laws as well. Okay. Now based on this dihybrid cross, Mendel gave another law known as law of independent assortment. Law of independent assortment tells us that the segregation of alleles of one gene in no way interferes with the segregation of alleles of the other gene. So both of them happen independently of each other. So here we can see the process of independent assortment taking place. So this law was given as a result of your dihybrid crosses. So till now what we have seen is one gene is responsible for one particular character. But in case of pleiotrophy, there is a bit sort of difference. What we see here is a deviation. Here, one gene is responsible for more than one character. For more than one character. So pleiotrophy can be seen when one gene not only regulates one character but also multiple characters. So an example for these is the disease phenylketonuria. In the disease phenylketonuria, phenylalanine, which is an essential amino acid that we get from our diet, is usually hydrolyzed into tyrosine, another amino acid. Now tyrosine is involved in the synthesis of neurotransmitters, right? So phenylalanine is an essential amino acid that we take from our diet, and it is readily converted to tyrosine in the normal physiological state in our body. This reaction is catalyzed by the enzyme PAH. PAH. Okay. So in phenylketonuria, the gene responsible for producing PAH is defective. As a result, this particular reaction does not proceed. There is no conversion of phenylalanine into tyrosine. Rather, this reaction stops as there is a deficiency of the enzyme involved. As a result, there is accumulation of phenylalanine within your brain. Okay, phenylalanine sort of increases and accumulates within the brain. And because this particular amino acid is also poorly absorbed by the kidney, it is readily excreted in the urine itself. So it can easily be detected from testing the urine levels. Now, the gene that is responsible for producing PAH is a pleiotrophic genes. So the individuals, those who are affected with phenylketonuria, they not only develop an accumulation of phenylalanine leading to mental retardation in the brain and poor excretion by the kidneys, but there is also multiple effects of this gene. People who have phenylketonuria also show reduced pigmentation. They also show reduced pigmentation of hair and skin. So people who have phenylketonuria, this particular gene, that is the gene responsible for producing PAH enzyme, is involved in multiple pathways and is responsible for more than one trait. That is why in people suffering from phenylketonuria, we see a variety of symptoms or disorders. Okay, so phenylketonuria is an example of pleiotrophy. Okay. Now something very, very exactly antagonistic to this is another mechanism known as polygenic inheritance. Now in pleiotrophy, what we had seen is one gene was responsible for more than one trait, whereas in polygenic inheritance, poly means many, right? So it's polygenic. Many genes together are responsible for the expression of one trait. So here, one trait is regulated, is influenced by many genes. Such an inheritance is known as polygenic inheritance. And such traits that show polygenic inheritance are known as quantitative characters. Quantitative traits or characters. And they are also known as continuous traits. They are also known as continuous traits. Examples include skin color. Skin color in humans that shows a gradient, whereas also your blood pressure, weight, height, and also milk production in cattle. So these are certain examples of polygenic inheritance. So skin color, blood pressure, weight, height, milk production, all of these are examples of polygenic inheritance. All these characters, they are known as quantitative traits because they can be quantified. And if you draw a graph for polygenic inheritance or for quantitative traits, they will show a bell-shaped curve. They will show a bell-shaped curve. Now, unlike the Mendelian traits, for example, Mendelian character like height was either the particular individual was tall or short. But here we can see a complete gradient. We can see a complete gradient for the quantitative traits. There are no discrete sort of traits here, but rather it is quantitative and continuous. Let's take an example to understand this better. Skin color. Skin color is influenced by 3 genes A, B, C. There are 3 genes that regulate the inheritance of skin color. Now at each of the locus there is 3 possible genotypes that can be possible. Just to give you an example, like in A gene can be homozygous dominant, it can be heterozygous, or it can be homozygous recessive. So similarly, at all the 3 locus, different genotypes are possible. As a result, we get a lot more variation. We see a lot of variation due to the different genotypes at different locus. When all the genes are in homozygous dominant condition, meaning all the genes are expressing themselves, they are producing proteins, enzymes required, in that condition, the pigmentation will be at its maximum. So here, there will be the darkest skin color possible. Now what about when it is heterozygote? When all the genes are in heterozygous condition. This will result in intermediate skin color. And when all the genes are homozygous, then we get the lightest form of pigmentation possible, that is, the person will be very fair. So that was an example of polygenic inheritance. We studied certain examples and we learned about certain characteristics of polygenic inheritance. So what we have studied till far is Mendelian genetics, right? Mendelian genetics. But there are also genes or traits that deviate significantly from the ratios that Mendel gave. Such genes are said to follow non-Mendelian inheritance. And we study them under non-Mendelian genetics. So under non-Mendelian genetics, the first phenomena that we have is incomplete dominance. What is complete dominance? We say a particular gene is completely dominant when, in the heterozygous condition, the dominant allele expresses itself while the recessive one is not able to express itself. So what happens in the case of incomplete dominance? In case of incomplete dominance, in a heterozygous condition, a novel phenotype is observed. We see a new novel phenotype in the heterozygous condition that does not resemble either of the homozygous condition. In the Mendel ratios, in the monohybrid cross that we studied, homozygous dominant and heterozygous both were giving me the same phenotype, that is tall, right? The phenotype showed in both the conditions was the same, but that is not in the condition in case of incomplete dominance. In incomplete dominance, the heterozygote condition will produce a novel, a new phenotype. An example of incomplete dominance is shown by the flowers of snapdragon. Let's study this with an example to understand it better. Okay, so in snapdragon flowers, there are two types of flowers. We have white and we have red as well. So in the parent generation, again we consider pure lines, homozygous. We take a plant that is red and has the genotype dominant homozygous, and we cross it with a plant that is recessive homozygous, that is white in color. So we are crossing a red flower and a white flower, both of which are homozygous dominant. The red one is homozygous dominant, whereas the recessive one is homozygous recessive. Now the F1 progeny that we get would again be heterozygous, very much similar to the Mendel cross that he performed. We would get a heterozygote, right? But what is different here is that the heterozygote, instead of being red, now it is showing a new phenotype that is pink. That is pink. Okay. So heterozygote shows a new characteristic that is pink. Now pink is a characteristic that is different from red and white. When we cross heterozygote, in the F2 progeny, what are we getting? In the F2 progeny, we get a red flower, we get two pink flowers and one white flower. So what's the ratio that you observe in the F2? In the F2, the ratio that we observe is, let's first talk about the genotypic ratio. The genotypic ratio is 1, 2:1, right? So the genotypic ratio is 1, 2:1. Very much similar to the monohybrid cross ratio, right? What about the phenotypic ratio? The phenotypic ratio here is 1:2:1, because 1 is red, 2 are pink, and 1 is white. Okay. In the monohybrid cross, the phenotypic ratio that we arrived was 3:1, but we see here a deviation from the classic phenotypic ratio given by Mendel. Here, the phenotypic ratio is resembling the genotypic ratio, another very important characteristic for an incomplete dominant cross. So in incomplete dominance, what have we seen? First of all, in a heterozygous condition, there would be a novel phenotype. Second of all, the genotypic and the phenotypic ratios would be similar. Let's move on to the second phenomenon, that is codominance. Okay. Codominance, as the name itself indicates, it is when in a heterozygote condition both the alleles will express themselves. Mendel said that in a heterozygous condition only one gene or one allele will be able to express themselves, while in case of codominance both the alleles are going to express themselves. An example for codominance is blood groups in humans. Now, blood groups in humans is controlled by the gene I. This I gene has three alleles: IA, IB, and I. Right? This can also be shown as IO. So normally all the genes that we have considered till now have two traits, but we see here that this particular gene has more than two alleles. So this shows multiple allelism. That is, it simply has more than two alleles. So I gene has three alleles. Now what is the function of these alleles? They code for the antigen that is to be added on the RBCs. Now depending on the allele, the antigen would be added on the RBCs. For example, the allele is IA, then the antigen that would be added would be A. If it's B, then it would be antigen B. And if it's O, then no antigen would be added onto the RBC surface. Your allele IA and IB, both of them are dominant over O. Meaning that when they are present along with O allele, then that particular allele is going to express itself. For example, here antigen A would be added, so the person would be having A blood group. Similarly for B as well. B is also completely dominant over the I recessive allele. Okay. So what would happen if both IA and IB are present in a heterozygous condition? If both IA and IB are present in a heterozygous condition, what we observe is on the RBCs now two types of antigen are added. There is antigen A as well as the sugar B. Both the antigens are added, so each allele is able to express itself, so they show codominance. Both the alleles are able to express themselves even in heterozygous condition. So in Mendelian cross, when we were discussing monohybrid cross, there we had seen that both homozygous as well as heterozygous genotype is going to give me the same phenotype, that is tall. So how was Mendel able to differentiate which phenotype is which? Like if the unknown plant is showing tall phenotype, how would I know its genotype? It can either be heterozygous or homozygous. For this, Mendel performed test cross. In this particular cross, you are testing whether the unknown plant that is given to you is homozygous or heterozygous. So in a test cross, the unknown plant is crossed with a homozygous recessive plant. Homozygous recessive . So the unknown plant is crossed with a homozygous recessive plant of the given trait. Now, if the unknown plant is homozygous, then all the progeny produced will show one single phenotype, right? All of them, 100%, that is, all of them would be showing one single dominant phenotype. Right? Whereas, if the unknown plant was heterozygous, in the resulting test cross, the progeny that would be produced would be in 1:1 ratio. Would be in 1:1 ratio. So depending on the progeny that we are getting, we can say whether the unknown plant was homozygous or heterozygous. Okay, so Mendel, after he gave this concept of genes and factors, later that was the same experiments were conducted and performed by other scientists, and they came to realize how important the work of genetics that Mendel performed was. Later, Boveri and Sutton gave the chromosomal theory of inheritance. Boveri and Sutton were studying cell division when they found that the movement of chromosomes is very much parallel to the behavior of genes. That is, as chromosomes move, along with them genes are moving. When they pair, along with them the alleles of a gene are also pairing. And when they segregate during gamete formation, basically your alleles are also segregating. So this was the chromosomal theory of inheritance given by Boveri and Sutton. So basically, by this time, we were able to allocate the fact that these genes, these factors, they are located within the chromosomes. Now, the alleles of a particular gene, they are located on homologous chromosomes. Homologous chromosomes are the ones that form a pair in us. We have 23 pair of chromosomes, right? So each pair is constituted by a homologous pair. Now in the homologous pair, each chromosome is received one from the father and the other from the mother, right? So we have two copies for a given gene, two alleles we can say, one from our father, other from our mother. Now let's come to the concept of linkage. The concept of linkage was given by Thomas Hunt Morgan. He was studying Drosophila, what we call as the fruit flies, Drosophila melanogaster. Now Thomas Hunt Morgan was studying X-linked traits in Drosophila when he stumbled upon linkage. Now what is linkage? Linkage is the physical association between two genes that are found on the same chromosomes and they are located very close to each other. When two genes, they are located very close to each other, there are chances that they may be inherited as a unit, right? The physical association shown by these two genes is what is known as linkage. Linkage is shown by genes that are present on the same chromosome, and within the same chromosomes they are very, very close to each other, right? So when they are close to each other, they are inherited as a unit. You might have learned about crossing over in meiosis. So I've drawn a chiasmata here. So during meiosis, when crossing over happens between non-sister chromatids, so here you can see two chromatids that are taking place in crossing over, right? And as a result of crossing over, there is genetic exchange leading to recombination. So basically, the two non-sister chromatids that participate in crossing over leads to exchange of information between these two sister chromatids, and as a result, there is recombination that takes place. New combinations or new variations are observed within the progeny, within the gametes, because of these crossing over. So, if two genes are located very close to each other, the chances of a crossing over happening between them is very low, right? So there the chances of recombination is very, very less. So more close to particular genes are within a chromosomes, the less there would be chances of recombination between them. Okay. This was applied by Alfred, a student of Thomas and Morgan, to develop maps. So he basically used the recombination frequency between two genes to map them out on a chromosome. If the recombination frequency between two genes were very, very less, that means they are present on the same chromosome, and if they are close to zero, that is 100% linkage can be observed in that condition. Let's now move to pedigree analysis. So in humans, we can study the inheritance of a particular trait or disorder with the help of drawing a family tree and analyzing this. That is simply referred to as pedigree analysis. So in pedigree analysis, we are trying to find what is the inheritance pattern of a particular trait or disease. So there are certain symbols that we use in case of pedigree analysis. The males are usually represented with a square, whereas the females are represented with a circle, and a mating between both is represented by a line drawn horizontally connecting both of them. Progeny are denoted by drawing a vertical line connecting to the progeny. The affected individuals, or the individuals that possess a particular trait, are often shaded and highlighted. Like in this condition, in the first generation, the female is affected, and then in the second generation also, the female is affected. The rest that are not shaded are unaffected individuals. So in pedigree analysis, let's first focus on autosomal recessive disorders. What are autosomal recessive disorders? These are the disorders that are only expressed when the individual is homozygous recessive for that particular trait or disorder. Only when the individual is homozygous recessive will they be showing that particular trait, whereas an individual that is heterozygous is going to act as a carrier. They will not exhibit that particular symptom, that particular disease or trait, but they are going to carry it forward to the next progeny. So they are known as carriers. Now, because it is autosomal, we know that in humans, we have 23 pair of chromosomes. Out of these 23 pairs, 22 are autosomal, while the 23rd pair is a sex chromosomes, right? So, if the gene or the trait associated with a particular character, trait, or disease is located in autosomes, we call them as autosomal recessive disorder, right? Autosomal. So how do I know? By referring to a particular pedigree whether that particular trait is autosomal or it is sex-linked, whether it is dominant or recessive, let's see. So how do I know whether a given disease is autosomal or X-linked? We can simply see and analyze the pedigree. If the number of males and females affected are nearly equal, then that is an autosomal disorder. as you can see, here is one female that is affected, and here we have one male. So number of progenies that are affected, male and female ratio, is equal. So we say that this is an autosomal disorder. Now how do I know that this is a recessive disorder as well? The biggest hint is here. In a recessive disorder, the trait or the disease is going to skip generations. You can see, I cannot see that particular disease in the first generation, right? So a trait or a disease that skips generation is recessive. Another clue is that the affected progeny is going to have unaffected parents. So when the affected progeny have unaffected parents, we can say that the given trait is recessive in nature. Next, let's look at some of the autosomal recessive disorders. Sickle cell anemia is an example of autosomal recessive disorder. In sickle cell anemia, the RBCs, they adopt a sickle cell shape under low oxygen conditions. So here we have two alleles, HBA and HBS, right? HBA and HBS. These both alleles, HBA is the normal allele, whereas HBS is the mutant allele. When an individual is homozygous for the HBS allele, then that individual gets sickle cell anemia. So this particular disorder can only result when that particular individual is homozygous for the given allele. And when we analyze and compare the gene sequence of a normal allele and a mutant allele, what we observe is a change or a mutation in the gene itself. So in the genes, there is one single base pair substitution that has taken place. A single base, adenine, was changed to thymine. As a result of this single substitution, what happens is, in the beta chain protein that we are getting, in the 6th position, where naturally glutamic acid was present, now as a result of the mutation we are getting valine. Okay. So this is the molecular basis behind your sickle cell anemia. So in sickle cell anemia, there is a single amino acid change that is resulting in the 6th position as a result of single base substitution. Let's move on to a second disorder, that is thalassemia. Thalassemia is again an autosomal recessive disorder. Now, thalassemia is of two types. Okay, in case of thalassemia, let's understand first a bit about hemoglobin first, then we'll come to thalassemia. Hemoglobin, the name itself, hemoglobin, is made of two components, heme, the iron part, and globin, the protein part. So hemoglobin consisted of heme, that is iron, and a protein part. The protein part of hemoglobin is made of alpha and beta chain. So we require alpha and beta in order to form the protein component of hemoglobin. Now, there can be defect in either the alpha chain or the beta chain, right? There, in case of thalassemia, the alpha chain or the beta chain is reduced or absent. Their production is reduced or absent. So depending on which chain it's affected, thalassemia can be classified into two types. Alpha thalassemia and your beta thalassemia. Alpha thalassemia can result as a result of 2 genes that are responsible for production of alpha genes. HbA1 and HbA2. Both of them are located on the 16th chromosome. Both of them are located in the 16th chromosome. So HbA1 and HbA2 are two genes that are responsible for production of the alpha chain that are located in the 16th chromosome. So if there is any mutation in any of the alleles, now the 4 alleles, there are 2 genes and we have 2 alleles of each gene. So there are 4 alleles. So depending on the severity of mutation, the person is affected. Alpha chains will either be severely reduced, the production of alpha chains can be severely reduced, or they may be absent altogether. Now let's move to beta thalassemia. In case of beta thalassemia, the gene affected is HBB. HBB gene is affected and this gene is located in 11th chromosome. This gene is located in the 11th chromosome. So again, if this gene is affected, there would be reduced production of beta chain resulting in beta thalassemia. So that was about thalassemia. Another example of autosomal recessive disorder is phenylketonuria. Phenylketonuria, we had already discussed in depth about phenylketonuria, so I'm not going to repeat that out here. Let's move on to X-linked disorders. So X-linked disorders. Now these are the traits or disorders that are linked with the X chromosome. That's the reason we are calling them as X-linked traits or X-linked disorders, right? The ones that we have to learn in your NCERT is X-linked recessive disorders, now in case of autosomal disorders, males and females, they are equally affected. But whereas in the case of X-linked disorders, there is a difference. We know that in case of females, we have 2 X chromosomes, whereas in case of males, they only have 1 X chromosome. The other one is a Y chromosome. So I can say, with respect to X chromosomes, males are hemizygous. Now because females have 2 copies of the X chromosome, for them to be affected by a recessive disorder, both the alleles should be recessive. Both the alleles should be recessive, then only will the female be affected. Now what about in case of males? In case of males, even if only they have just 1 copy of that recessive allele, it is going to express itself. Even if there is just 1 copy of that recessive allele, they are going to express that. As a result of this hemizygous condition, males are more often more affected. They are affected more for an X-linked recessive disorders. Right? The chances of males getting affected by an X-linked recessive disorder is higher than that of females. So let's study the 2 examples of this as well. So the first disorder is hemophilia. Hemophilia is also known as the royal disease. Okay. Hemophilia is a clotting disorder. In this, the clotting proteins that are involved in the clotting cascade is affected. And which clotting protein? Factor 8. Factor 8 is affected in case of hemophilia. So as a result, clotting does not happen. So the individuals who are hemophilic, even if they get a paper cut or a nosebleed, there's a high probability they may even die from that resulting injury. So this is an X-linked recessive disorder, meaning that the females will only be affected when they are recessive for this particular, both homozygous recessive for this particular trait, whereas the males will get affected even if they have just one copy of that recessive allele. Now females can act as the carriers here. Females can act as the carriers here. They will not show the disease, but they have one copy of the allele that they can pass on to their progeny. Queen Victoria was the carrier. She brought that disease along with herself to the royal family. Okay, males can never be carriers. Just one thing that I would like to remind you here is males can never be carriers because if they have even one copy, they are going to express themselves. Let's move on to the second disorder, that is color blindness. Color blindness is again an example of X-linked recessive disorder. So in our retina, for the perception of light, there are different photoreceptor cells. These photoreceptor cells are your rods and cones. Rods are responsible for vision in dim light or night vision, whereas your cones are responsible for color vision. They are the ones that are responsible for color vision. Now, depending on the wavelength of light they're sensitive to, cones are of three types: blue, red, and green. Right? Under your X-linked recessive disorder, though there are only two types that we study, that is green and red color blindness. Because the blue one is that is responsible for color blindness is located in an autosomal particular gene. So for an X-linked disorder, we only study green and red under this. Okay, so people who are colorblind, they cannot differentiate between colors. They see the world as a contrast of gray. So again, similarly here, for a female to be affected, she will need to have two copies of the recessive allele, whereas for a male, they need just one copy to express that particular trait or disease. So as a result, the probability of them expressing this disease is far way greater than that of a female. If you look at the ratios, it's 0.4 for females, whereas it's 8% for males. Now we looked at some of the disorders caused by X-linked disorders, X-linked recessive disorders, as well as autosomal recessive disorders. Now let's look at chromosomal disorders. Chromosomal disorders, they result when during meiosis, there is improper cell division. So as a result, during meiosis, either one extra chromosome was added or one chromosome was reducted from the total genome, right? So we can either have aneuploidy. Aneuploidy occurs when extra chromosome is either added or subtracted, right? Then we have polyploidy. Polyploidy occurs when a whole set is either added on or deleted from the genome of the gamete, right? So either we can have polyploidy, aneuploidy. The disorders that we see in humans are a result of aneuploidy. In plants, polyploidy is common, often seen. Okay? In aneuploidy, we have autosomal as well as sex chromosomes affected aneuploidy. Okay, so the first disorder that we study under this is Down syndrome. Down syndrome results as a trisomy of 21st chromosome. 21st chromosome is an autosomal chromosome. Now, as a result of trisomy of the 21st chromosome, we get Down syndrome. So in this disorder, the individuals that have Down syndrome, they have mental retardation, their physical growth isn't that proper, they have psychomotor disabilities as well. So And they have special characteristic facial features as well. They have a partial open mouth, their eyes are smaller, right? There are creases in their palms, and they often have congenital heart diseases. Now let's look at the second disease, that is Klinefelter's. Klinefelter syndrome results due to an additional X copy. Now, the people who have Klinefelter syndrome, they are basically males. They are males, so they are going to have XY, right? Now, in addition to the XY they also have an additional X. So that makes it XXY. And because they have one extra chromosome, they have 47 chromosomes in total. Okay. So people who have Klinefelter syndrome, they are basically males who have feminine characters and show gynecomastia. That is the development of breasts. Okay. Third, we have Turner syndrome. The people who show Turner syndrome are basically females. In them, what happens is one X is missing. So instead of XX in the sex chromosome constitution, they have an X and an O. As a result, they have just 45 chromosomes instead of 46. So here, the females that are resulting from Turner syndrome, these are females, but they have rudimentary gonads, as a result of which they are sterile, and the other feminine characters are not well pronounced or are lacking. Okay. So with this, we have went over majority of the topics and we have covered them. So if there is any more chapters that you want to study with me and do revision classes of that, do let me know in the comments. Okay, so we have completed genetics, one short revision. So see you in the next class.